Welcome to the deep dive. Today we're taking a, well, unnecessarily sober, but hopefully very informative look at recent advances in advanced prostate cancer research.
Mm.
We've gathered quite a bit of material research findings, articles to help you make sense of the key developments.
Absolutely. Our goal here is really to distill the most important information from these sources. We want to help you understand what these new findings mean, particularly for treating metastatic castration-resistant prostate cancer, or mCRPC, and also metastatic hormone-sensitive prostate cancer, MHSPC.
Right, and we'll treat each source sort of like a subject we're discussing together. We'll kick things off by looking at some promising clinical trial data, starting with the big Phase 3 results and working down to earlier studies.
And then we'll shift focus a bit, digging into some really exciting discoveries happening more at the lab bench level, the preclinical stuff, especially around how prostate cancer spreads to bone, which is, a major challenge.
It's a field that moves fast,
Yeah.
so these sources really give us a valuable snapshot.
They do. Shows where things are heading.
Okay, let's unpack this, starting with those clinical trial findings. Phase 3 trials. First, this is really the highest level of evidence. First up, the ENZAMET trial.
Right, ENZAMET. This focused on MHSPC, so metastatic hormone-sensitive prostate cancer. It was comparing adding enzalutamide, that's an androgen receptor inhibitor, to standard testosterone suppression, sometimes with early chemo, docetaxel.
Compared to what?
Compared to the older standard, which was using non-steroidal anti-androgens, NSAAS, plus that same testosterone suppression.
And the big news here, after a pretty long median follow-up, 98 months, enzalutamide significantly boosted overall survival, OS.
That's the headline, really. A big difference. Median OS was 95 months for the enzalutamide group compared to 70 months for the NSAA group.
Wow, that's more than 2 years difference in the median.
Exactly. And looking at it another way, at the 8-year mark, half the patients, 50% on enzalutamide, were still alive, versus 40% for the standard care group. It works out to about a 27% reduction in the risk of death. That's potentially years of life.
Really significant. And I saw it also helped with delaying the cancer worsening. Clinical progression-free survival, PFS.
Yes, big benefit there too. A 51% lower risk of the cancer progressing or death. And importantly, fewer deaths were actually caused by the prostate cancer itself in the enzalutamide arm.
And was there anything predictive? Like, could doctors tell early on who might do better?
Yes, that was interesting. The study found that getting a really low PSA level, 0.2 nanograms per milliliter, or even lower just 7 months into treatment, was a very strong predictor of better long-term survival. Didn't matter which group you were in initially, it's a useful early signpost for clinicians.
What about side effects, though? Enzalutamide for, what, nearly 5 years on average?
Yeah, the average time on enzalutamide was 58 months versus 36 for the NSAAS. But the source indicates the safety profile was manageable. Despite that much longer duration, there wasn't a significant jump in severe toxicity.
Any limitations mentioned?
The main one noted was needing more detailed data on what happened after the trial, the subsequent therapies patients got. That can obviously influence overall survival too, so it's something to keep in mind.
Okay, makes sense. Let's switch to another big Phase 3 trial, Talazoparib. This one's in the mCRPC setting, metastatic castration-resistant.
Correct. Talazoparib looked at mCRPC patients who hadn't had prior life-prolonging therapy for this stage of the disease. It tested adding Talazoparib, that's a PARP inhibitor, to enzalutamide, versus just using a placebo with enzalutamide.
And importantly, it included all comers, right? Not just patients with specific gene mutations.
Exactly. They didn't pre-select for HRR mutations like BRCA1 or 2, though they did look at that subgroup later.
And the final analysis confirmed an overall survival benefit for the combination.
Yes, it did. Median OS was 45.8 months with Talazoparib plus enzalutamide compared to 37.0 months for placebo plus enzalutamide. It was statistically significant, representing about a 20% reduction in the risk of death overall, another win for combination therapy.
And they also reconfirmed the radiographic progression-free survival, rPFS, benefit.
They did. That was 33.1 months for the combo versus 19.5 for the control. So substantially longer time before the cancer showed progression on scans.
Was the benefit stronger in certain groups? Like those HRR mutated patients?
It was, yes. For patients with HRR mutations like BRCA1, BRCA2, ATM, the benefit was even more pronounced. The risk of death was cut almost in half, a hazard ratio around 0.55.
But there might still be some benefit even without those mutations.
Well, the exploratory analyses hinted at that possibility. It wasn't the primary finding for that group, but it suggested there might be some benefit even in the HRR non-mutated population. That's still being looked into, understanding exactly who benefits most.
And safety for Talapro too, with the longer follow-up.
No new major concerns popped up. The most common severe side effects, grade 3 or higher, were blood-related. Anemia in 49% and neutropenia, low white cells, in 19%. About 22% did stop Talazoparib due to side effects, but overall it was seen as manageable.
Okay, so those are two major phase 3 updates.
Yeah.
Let's move to phase 2 trials. Smaller studies, but often looking at interesting new combinations or specific situations. The COMRADE trial focused on bone mets and mCRPC.
Right, COMRADE. This was interesting. It randomized patients with mCRPC and bone metastases 1-to-1, either combination therapy, Olaparib, another PARP inhibitor, plus radium-223, which is that targeted alpha therapy for bone.
Or just radium-223 alone.
Exactly. And the key finding was that the combination significantly improved radiographic progression-free survival, the rPFS.
How much better?
Median rPFS was 8.6 months for the combination versus 4.0 months for radium-223 by itself. So more than double the time until progression was seen on scans. That's quite meaningful for patients with bone-dominant disease.
And did that benefit depend on having those HRR gene mutations?
Interestingly, no. The source stated the benefit was seen regardless of HRR status. That's different from how we usually think about PARP inhibitors working primarily in HRR-deficient cancers. It suggests some kind of synergy with the radium-223 that goes beyond just DNA repair defects.
Any other benefits noted in COMRADE?
Yeah, a couple of secondary things. Longer time until the patient needed the next line of therapy, 12 months versus about 8. And fewer symptomatic skeletal events, like fractures or needing radiation for bone pain, at the 1-year mark.
Overall survival.
Similar at the 12-month point, according to the source. But the primary goal, delaying progression, was definitely met. Safety was considered manageable, although, as you'd expect with two active drugs, severe side effects were more common in the combination arm.
Got it. Another phase 2 update involved Mevrometostat.
Yes, Mevrometostat. This trial looked at mCRPC patients who'd already progressed after abiraterone and hadn't had much chemo. It compared adding Mevrometostat, which is an EZH2 inhibitor, a different mechanism to enzalutamide and standard ADT, versus just enzalutamide and ADT.
And again, a significant improvement in radiographic progression-free survival for the combination.
Absolutely. A big difference here. Median rPFS was 14.3 months for the combination, compared to just 6.2 months for enzalutamide alone. That's more than doubling the rPFS in patients already resistant to one androgen pathway drug.
Were there improvements in other measures like tumor shrinkage or PSA response?
Yes, the source highlighted those too. For patients with measurable tumors, the objective response rate, ORR, was higher, 26.7% versus 14.3%. And more patients had a significant PSA drop, a PSA 50 decline, 34.1% versus 15.4%.
So clear signs of anti-cancer activity.
No.
Safe.
Manageable adverse event profile overall. But again, like COMRADE, the combination arm did see a higher rate of severe adverse events, about 54% versus 43%. It just underscores that when you combine therapies, managing side effects becomes even more crucial.
So we've hit the big phase 3 shifts and some key phase 2 combinations. But wow, the pipeline seems packed with earlier phase studies exploring totally new angles, right?
Oh, absolutely. It's incredibly active. Researchers are looking at completely different ways to attack the cancer. For example, there's a whole class of novel radioligand therapies. We saw updates on things like Lu-177, NYM032, and Terbium-161 PSMAIT.
These are like next-gen versions of drugs like Pluvicto.
Kind of. They use isotopes like lutetium-177 or even terbium-161, which has unique properties, and link them to molecules that specifically target PSMA, that protein often found on prostate cancer cells. The idea is to deliver targeted radiation right to the tumor. Early data looks promising for both activity and safety.
And then there are antibody-drug conjugates, ADCs.
Exactly. Think of them as, well, smart bombs. An antibody finds a target on the cancer cell like B7H3, which MHB088C targets, and delivers a really potent chemotherapy payload directly. The payload in MHB08C, called super topoi, is noted as being very powerful.
And did that show promise?
Early results in heavily pretreated patients looked encouraging, a disease control rate of over 95% in those with measurable disease, and good 6-month rPFS. It suggests this approach can work even after many other treatments have failed.
We're also seeing totally different mechanisms. PT112 got FDA clearance for phase 3.
Yeah, PT112 is quite different. It's a small molecule, a pyrophosphate conjugate. It messes with the cancer cell's ability to make proteins, inhibits ribosomal biogenesis. But it doesn't just kill the cell. It seems to do it in a way that elutes the immune system, causing immunogenic cell death.
And it likes bone.
That's the other key part. It has osteotropism, it concentrates in bone tissue, so very relevant for prostate cancer. Early phase 1 results in heavily pretreated patients showed some durable responses, even a few patients surviving 2 years or more. Safety looked manageable too.
And immunotherapies are evolving too. Pasritamig.
Right, Pasritamig, or JNJ78278343. This is a T-cell redirector. It targets KLK2, human kallikrein-2, which is very specific to prostate tissue, including cancer.
How does it work?
It acts like a bridge. One end grabs onto KLK2 on the cancer cell, the other end grabs onto CD3 on a T cell, basically dragging the T cell over to kill the cancer cell. The phase 1 results showed it was safe and had clear activity, PSA responses, even tumor shrinkage in a very heavily pretreated group. It really validates KLK2 as a target.
And there was also a study combining an existing radioligand at Lu-177 with an immunotherapy drug, Gotistobart. That's Preserve 006.
Yeah, that's interesting. Gotistobart targets CTLA4, an immune checkpoint. The idea was maybe adding this immunotherapy could overcome immune suppression within the tumor, making the Lu-177 radiation work even better.
Get it.
The early phase 1 data suggested yes. The PSA 50 response rates were significantly higher in the combination arms compared to Lu-177 alone. It hints that combining targeted radiation with immunotherapy could be a powerful strategy, possibly overcoming resistance.
So pulling back from the specific drugs, the theme across these earlier studies seems to be innovation, new targets, smarter delivery like ADCs and radioligands, and finding new ways to leverage the immune system.
Exactly. And often showing promise in patients who have run out of standard options. It really shows the breadth of the research effort.
Okay, before we dive into the preclinical world, let's quickly touch on a real-world evidence study. This one looked at Sipuleucel-T.
Right, Sipuleucel-T, that's the immunotherapy, the vaccine basically, made from a patient's own immune cells trained to target PSA. It's been approved for a while. This study used a large database, SONR, to see how patients getting it in routine practice fared compared to those who didn't in the MCRPC setting.
And the top-line result looked pretty dramatic, right? Significantly longer survival for the Sipuleucel-T group.
It did. The median overall survival reported was 44 months for those who got Sipuleucel-T versus 24 months for the comparison group in the database. And at 5 years, 28% of Sipuleucel-T patients were alive versus 12% of the others. That translated to a reported 70% lower risk of death.
Those are big numbers.
They are. Very encouraging on the surface. But, and this is crucial, the source itself rightly points out the big limitations of this kind of retrospective real-world data.
Because patients weren't randomly assigned.
Exactly. There's a huge potential for selection bias. The study noted that the patients who actually received Sipuleucel-T tended to be younger and have lower PSA levels to start with.
Meaning they might have had less advanced disease anyway.
Precisely. That alone could account for a significant part of the survival difference. The study couldn't adjust for everything: other health problems, specific prior treatments like abiraterone or enzalutamide, which weren't fully captured, socioeconomic factors.
So it shows an association, but you can't definitively say Sipuleucel-T caused that entire survival benefit.
Correct. It highlights the challenge of interpreting real-world evidence. It's valuable for seeing patterns in practice, but you have to be really cautious about drawing strong causal conclusions compared to a randomized controlled trial where groups are balanced from the start.
Okay, that's a really important caveat. Let's shift gears now to the preclinical side. This is the foundational lab work, often using cells or animal models, that helps us understand how prostate cancer works, especially its spread to bone, and uncovers completely new targets for future drugs.
This is where we get into the basic biology, trying to figure out the mechanisms. First up, some findings about a protein called ACBP. Using tools like CRISPR gene editing, scientists identified ACBP acyl coenzyme, a binding protein, as potentially a key driver of metastasis, specifically to bone.
What did the lab studies show?
In mouse models, higher levels of ACBP correlated with more cancer spreading to the bones from lung or breast cancer models initially. And looking at human genetic data seemed to support this link between ACBP, bone metastasis, and unfortunately, poorer survival.
And how does ACBP actually help the cancer spread to bone? What's its job?
It seems to help the cancer cells generate energy through a process called fatty acid oxidation, or FAO, basically providing fuel for the demanding process of setting up shop in the bone. It also helps the cancer cells avoid a specific type of cell death called ferroptosis.
So it fuels them and protects them.
Yeah.
Did they try blocking it?
Yes, in the mouse models, they tested compounds, one called IKE that promotes that ferroptosis cell death, and another called inamoxer that interferes with the FAO fuel production. Both approaches inhibited bone metastasis in the mice.
Suggesting potential treatment strategies.
Potentially, yes. Targeting the cancer's energy supply or making it vulnerable to ferroptosis could be avenues for clinical development down the line.
Very interesting, targeting fuel and defense. Okay, another key protein highlighted in preclinical work, ASH1L.
ASH1L. This one was identified as being crucial for kind of priming cancer cells to invade bone in the first place. They found it's often amplified genetically or overexpressed in metastatic prostate cancer, and again, higher levels linked to more advanced disease and worse outcomes.
How do they pin down its role?
Through direct experiments. If they reduced ASH1L levels in aggressive prostate cancer cells in the lab, the cells became less invasive. And when they used CRISPR to completely remove ASH1L in mice, it dramatically suppressed bone spread and significantly improved survival.
And making more ASH1L had the opposite effects.
Exactly. Increasing ASH1L boosted bone tumor growth in mice. It seems like a pretty clear driver based on this preclinical work.
What's the mechanism here? How does ASH1L prime cells for bone?
It's an epigenetic driver. ASH1L is a histone methyltransferase. Think of epigenetics like bookmarks and highlights on your DNA, not changing the words, but changing which parts get read. ASH1L works with another factor, HIF1 alpha, to switch on genes that promote metastasis by changing these epigenetic tags.
And does it only affect the cancer cell itself?
No, and this is really interesting. It also seems to change the bone environment. It helps reprogram immune cells, called monocytes, turning them into a specific type of tumor-associated macrophage, or TAM, that actually supports tumor growth.
Wow, so it makes the cancer cell more aggressive and makes the neighborhood more welcoming.
That's a good way to put it. It's a dual effect. And they did test targeting it. An inhibitor called AS99 suppressed bone mets and prolonged survival in mice. Blocking those reprogrammed macrophages, or inhibiting HIF1 alpha, also showed promise in mouse models. So ASH1L and its network look like very exciting potential targets.
Okay, one more piece of preclinical insight. This one used single-cell RNA sequencing to look at the immune cells inside actual bone metastases.
Right. This was a deep dive into the immune microenvironment of bone mets. They analyzed 42 samples from different cancer types, including breast and kidney, looking cell by cell. The really striking finding was that bone metastases aren't all the same immunologically. They seem to fall into 3 distinct immune patterns, or archetypes.
3 different immune flavors inside the bone tumors.
Pretty much. One type was rich in macrophages and osteoclasts. Those are the cells that break down bone. Another type was dominated by regulatory T cells and exhausted T cells that tend to put the brakes on an anti-cancer immune response. And the third type was characterized by lots of monocytes.
And these types didn't necessarily match the original cancer. Like, all breast cancer bone mets weren't the same.
Exactly. Bone mets from the same primary cancer could fall into different immune categories, while bone mets from different primary cancers could sometimes share the same immune profile. It suggests the local bone environment strongly shapes the immune landscape once the cancer gets there.
What does that mean for treatment?
Huge implications. It suggests, for instance, that therapies targeting osteoclasts might only work well in patients with that first archetype. Patients whose bone mets are full of exhausted T cells might be much better candidates for immunotherapies designed to, wake those T cells up.
So it really points towards needing to understand the specific immune makeup of the metastasis itself, not just the primary tumor type, to choose the best therapy.
Precisely. The study even identified potential new drug targets specific to each immune archetype. It paves the way for potentially much more personalized treatment strategies for bone metastases in the future.
Okay, let's try to connect these dots. On one hand, we have the clinical trial results showing real progress. New combinations like enzalutamide earlier on or Talazoparib plus enzalutamide later improving survival.
Right. And those phase 2 and earlier studies showing promise for completely new approaches, next-gen radioligands, ADCS, drugs hitting novel pathways like PT112, T-cell redirectors like Pasritamig, combining radiation and immunotherapy, tangible steps forward.
And then complementing that, we have this fundamental preclinical research, identifying proteins like ACBP that fuel cancer in the bone, or ASH1L that primes cells for invasion and even changes the bone neighborhood.
And that work showing the surprising diversity within the bone metastases themselves,
Yeah.
those different immune archetypes. It helps us understand why things happen and where new vulnerabilities might lie.
It really feels like progress is happening on all fronts, understanding the basic science of metastasis, especially to bone, and translating those insights, plus developing clever new therapeutic strategies into clinical testing.
It does. It paints a picture of moving towards more targeted, hopefully more effective, and potentially more personalized approaches for advanced prostate cancer, particularly when it involves the bones.
It's clearly a complex fight, but seeing the research advance like this, from the lab bench to phase 3 trials, is definitely encouraging.
Absolutely. Each study adds a piece to the puzzle.
So here's a final thought to leave you with, building on everything we've discussed. Think about this. How might understanding the specific environment within an individual's bone metastasis, maybe knowing its immune archetype from something like single-cell sequencing, eventually intersects with therapies targeting key drivers like ACBP or ASH1L, which we learned about from the preclinical work? Could we one day combine that deep biological knowledge of the specific metastasis with targeted drugs to create truly personalized treatment plans?
That's the hope, isn't it? Really tailoring therapy to the unique biology of the cancer right where it is. A powerful idea for the future.
Thanks for joining us for this deep dive. We hope keeping up with this research is valuable for you.
Yes, thanks for listening. The progress is undeniable.
Stay strong and fight on.